They're not in the cell cycle.
What is?
It just said what is zero.
Yeah, just know what it means. Know what it means. It means that the cell's not dividing.
So EG0.
Other questions?
What's the difference between a eukaryotic and prokaryotic gene structure?
Okay, so eukaryotic and prokaryotic gene structure. So everything that we've been talking about up until this point has been, we focused on eukaryotes because they're most relevant to us. But when you look at eukaryotic DNA, it's going to be linear and it's going to be double-stranded. And the way it's going to be arranged, you're going to have one promoter for one gene. Whereas in prokaryotes, they still have double-stranded DNA, but their DNA is circular instead of linear. So remember we said we can stretch ours out 100 whatever billion kajillion miles. Well, theirs is going to be a lot smaller and it's going to be circular in nature. And so remember we talked about the operons? The operons have, so if I just straighten this out just for simplicity, they're going to have one promoter regulating several different genes at a time. So those are kind of the main differences. The shape of the genome, one's linear, one's circular, and then the fact that in eukaryotes it's usually one promoter for one gene, whereas in prokaryotes it's one promoter. You use the operons, which control multiple genes with only one promoter.
So one promoter, they can, for a lot of different
For
genes.
multiple genes. Remember, we talked about the lac operon. We had one promoter that synthesized 3 genes that were involved in breaking down lactose. So that would be an example of a, bacterial genome.
Prokaryotes are linear.
No, eukaryotes are linear. Prokaryotes are circular.
I'm trying to find that in the material.
Yeah, we didn't really talk, the main thing I wanted you to know is we did talk about operons. And we also talked about eukaryotic promoters. So eukaryotic, we said, if there's a gene X, that means there's a promoter X. Whereas in prokaryotes, you can have a promoter X and have a gene X, a gene Y, and a gene Z.
Seeing how you just briefly talked about operons, can you describe again it says the lac operon functions to regulate the amount of lactose in the cell?
Mm-hmm.
Can you go over that?
So,
Dr. Gilly?
yep.
Before you get started, when you were talking about the extra credit, you said it's worth 40 points, right?
Yes.
Okay, is it like, are we getting graded out of the 40 points, or do we give 40 points,
You pretty much just get 40 points for doing it.
on what,
I'm gonna make a column in Blackboard. It's gonna be an extra credit column. So I'll put it there, so it's not gonna go to one specific place, but it will still add to your points. And then at the end of the semester, all those, volunteer participation points, I'm gonna add them into that column too.
So it's 40 out of 0, not 40 out of
It's 40 out of 0, not 40 out of 40. It's an extra credit. So you'll actually have over 100 or 1,000 points that you can get.
One of those.
So we're ready to do the lac operon? All right. So the lac operon,
What is that in that?
again, we have a promoter region. The promoter is where the replicating enzyme is going to bind, right? So this is where RNA polymerase is going to bind. And next to the promoter, we have an enhancer. I'm sorry, not enhancer region. We have an operator sequence. So that's the promoter. Sorry, that's the operator. This is the promoter. These are both non-coding, right? So they're not going to make protein. And so after the operator, that's when you have all of these genes. So let's just say, and I forget the names of the genes, but it's 3 genes. So let's call them gene 1, gene 2, and gene 3. So the purpose of the lac operon is to regulate the amount of lactose in the cell. Whenever there's no lactose present, Do we need the proteins necessary to break down lactose if there's no lactose present? No. Why waste the energy, right, to make all this protein? So when there's no lactose that's present, that means that these 3 genes whose sole job is to break down lactose, they're not needed. So we just turn them off. We're not making protein because we don't need them yet, because there's no lactose. So the way that we turn it off is because in the absence of lactose, there's going to be this suppressor protein. And that's going to bind to the operator sequence. And so what that means is, remember we got this big old RNA polymerase over here that's trying to go down and make the RNA, but if the suppressor's there, it's just in the way. He can't get around them. Now, if there is lactose present, that means it needs to be broken down, which means we need to turn this on so we can make these proteins that are going to break down lactose. So when lactose is present, do this again, so promoter, operator, gene 1, 2, and 3. So when there is lactose present, you have this suppressor that's still there, but lactose is actually inhibiting that suppressor. So where that suppressor would normally bind to the operator, basically the lactose has essentially blocked that sequence. It's basically preventing the suppressor from binding to the operator. But it only occurs, lactose is the only molecule that can inhibit the suppressor, or that can enable, or I guess negatively affect the suppressor molecule. So if lactose is present, it's always going to bind to the suppressor. If it's bound to the suppressor, that means the suppressor can't bind the operator. So that leaves the operator free, which means that RNA polymerase is going to go and be able to make those RNAs, which will eventually be transcribed into making those proteins that break down lactose. So when you turn on the pathway in the presence of lactose, you're making enzymes that break down lactose. So what are they going to do the minute they get made? They're going to go break down some lactose. All right? And then when they break down all the lactose, then it goes back to this stage. All right, so it's regulating the amount of lactose in the cell. If there's lactose present, then the operon's going to be turned on because you're making enzymes that break down lactose. If lactose isn't present, then there's nothing to break down, so there's no point in making a bunch of proteins to break it down if it's not around.
So the word, I think I was on the day you talked about operon, so the word operon is just those line of genes?
Yeah,
Turning them
this
on
is
and
just
off.
an example of an operon. But an operon in general is where you have one promoter
And gene.
responsible for replicating multiple genes.
Okay.
That's an operon.
Okay,
It's under 7.6.
okay, thank you.
And that would be called the suppressor, it's labeled on your repressor, so same thing.
Repressor,
Just making
suppressor.
sure.
Technically, yes, it's repressor,
Okay.
but if you put suppressor, I mean, it means the same thing.
Yeah, exactly, just making sure.
And some people call them inhibitors, so there's multiple different terms that can be used. So since your book uses repressor, I'll try to use repressor.
I'm with you, just want to make sure.
Yeah, Pat?
List of 3 functions of cell division. The one I wrote down was that it controls when the cell goes into mitosis.
That's what the G2 checkpoint does. The G2 checkpoint regulates when a cell goes from G2 into M phase, or into mitosis. Now the 3 functions of cell division, growth and development,
Tissue repair and
tissue repair, and reproduction. Anybody else? Yeah.
What two processes make up MPs? I know the different, like, I just don't know what 2 processes make up.
So mitosis and cytokinesis
Okay.
make up M phase.
You say M phase?
M phase. So when we're talking about M phase, we're always talking about the cell cycle. Like if we're talking about S phase, we're always talking about the cell cycle.
It just means all the stages of mitosis and.
All the stages of mitosis, so prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis.
You want us to write all that out for in-phase? Or just, what two processes?
If I say process,
Mitosis and-
mitosis is a process. I mean,
Okay.
technically prophase is a process, but I'll be specific.
Okay. Mitosis is,
Yeah, if I said what 2 stages, if I said what 6 stages make up in phase, but I'm not gonna do that to you. I'll say what, if there's a question on there, I'll say what 2 stages.
Cytokinesis, division of cytoplasm at that stage.
Mhm.
Mitosis is gonna be just cell replication.
It's not,
Sorry.
it's 2 processes. The whole thing of in phase is cell division. The first thing you do is you separate your DNA, which is what mitosis is.
Separate the-
Once you separate the DNA, then you separate the rest of the cell, which is what cytokinesis
And that's
is.
always last, yeah.
That's always last, yeah.
And you said that for the functions of cell division was reproduction, repair, and for growth?
Yeah, growth and development, tissue repair, and reproduction. So when we make our gametes, we're dividing our cell, right? So that's part of reproduction.
The difference between cytokinesis in plant and animal cells were the cleavage furrow and then the cell plate.
Yep.
Okay, great.
Explain how cancer and cell cycle are related.
All right, so the cell cycle regulates when a cell divides, right? So what is cancer?
Divided cell?
Uncontrolled cell growth. So in other words, unlike most normal happy cells that are in the G0 90% of the time, meaning they're not dividing, the cancer cells are going to be stuck in the cell cycle, so they're constantly going to be dividing. And if they're constantly dividing, that means they're making lots and lots and lots of copies of the cells, which is how you get a tumor. So a tumor is just uncontrolled cell growth, and the reason why it's uncontrolled is because it's going through the cell cycle more times than it should.
which that was, how the tumors.
How cancer is related to the cell cycle.
On the first section.
When you say how do tumors spread, is that when you're talking about masticize?
Yeah, when it metastasizes, so basically all I want you to know is you have a tumor here, and then a cell gets into the bloodstream and it goes elsewhere, gets deposited somewhere, and then makes a new tumor.
You got bacterial or viral? Yes, please.
Thank you for giving us a study guide.
Yes, thank you very much. Yeah.
Usually don't do that.
Crossing? Does crossing over happen in mitosis? No.
No,
Okay.
it only happens in prophase 1 and meiosis 4.
Okay.
Masticize?
When does the nucleolus disappear?
Oh, I know that, prometaphase.
Prophase.
Yeah.
According to Saga, it's prophase.
Prophase, it condenses,
And then
and
metaphase is disintegrates, so thank you.
In lab, they said they don't even want to use prometaphase. They said they're going to cut it down in a minute, you know. But
I wish,
how?
yeah, it makes it so much simpler. I don't know why they have to overcomplicate it.
Is it prometaphase?
I think there's some debate. Some sources say it's metaphase, or pro-metaphase, some say it's prophase.
What do you want?
I say,
Whatever you say.
What do you want?
so the DNA condenses and the nucleolus disappears. That's normally what happens in prophase.
Prophase.
Then in prometaphase, that's normally when the nuclear envelope starts to degrade. So I say prometaphase is when the nuclear envelope starts to disintegrate.
When the prometaphase, you want that?
I won't prometaphase. If I have a question, I won't put in an answer that'll try to confuse you. Like I won't put in prophase and prometaphase. I would put in like prometaphase, anaphase, and telophase or something, you know.
Primary fission?
We talked about that.
I didn't get it.
A quick question on the extra credit.
Mhm.
When we went to YouTube, there was only like 3 that you could access for free.
Really?
So you'll have a lot of the same,
That's fine.
So that's what I'm saying.
I mean, really, I'm gonna read like the first paragraph.
are you interested in giving an extension on it? I have mine, but some people may need an extension
Why?
because they didn't like the ones you could choose from.
That's not good enough. I'm giving you an easy 40 points.
More.
Easy
So
free
you also
ones.
have
You have to buy it.
I did not know that.
I ain't got $2.
$1.99.
They're like $1.99.
But there's also free ones.
Like three.
Yeah, there's free ones.
There's three
Yeah.
free ones? Well, the one that I happened to click on must have been the free one.
Yeah.
Is there a creator?
Most of them are just like clips of the video. You can watch like a 30-second clip.
they're all illegal download.
Yeah, I saw a bunch
I
of
watched
videos.
45-minute one, so I did that.
Yeah, I feel like.
I watched the 45 minutes. I want the extra points.
I paid for one.
DNA polymerase 1 is the one that stops.
I watched the one.
Polymerase 3 is gonna start and bind after the
After the primer gets made?
That was it.
After the primer gets made?
The polymerase 3 is gonna bind with the primer.
And that DNA polymerase 3 is what's actually making the DNA.
Okay, that's when it codes it.
Yeah,
Okay.
and so
That's what stop it.
the DNA polymerase
I
1,
wanna just say what stop it.
what that does is it takes out the RNA primers and then fills in the gaps left behind with DNA.
Are there any binary vision free?
Binary fission is just how a bacteria cell will divide itself into more bacteria.
It replicates its DNA, the DNA attaches to opposite sides, the cell elongates and eventually pinches in the two. Pretty straightforward.
Replicates, elongates, pinches into two.
So pinches will be on the cover?
Pinch, yeah, it's fission, pinch, separate, whatever. Something that tells me that it was one cell and now it's two cells.
What is the relationship between the original cell and the newly produced cell? Nothing.
In binary fission and in mitosis, you're making
Nothing. Exact
same copy.
A gene for mommy, gene for dad, you copy those. Perfect.
In meiosis, that's where you're making different genetic versions.
Meiosis, yes. DNA. A different copy.
Anybody else? That it? Alrighty. I will be around Friday from 10 to noon in the Science Learning Center.